Hello and welcome to this FTCOE webinar. Today we will hear from Dr. Fung, Professor at North Carolina State University, and Dr. Michelson, Professor at the Royal Melbourne Institute of Technology University, on physical characteristics of spatter stains on textiles. Without further delay, I will turn things over to our presenters.
Okay, thank you, Natalie, and thank you all for joining the webinar. And we also appreciate the opportunities provided by RTI for us to give the webinar. And today, Dr. Michelson and I will talk about physical characteristics on bloodstains on textile surfaces. So our topic belongs to the bloodstain pattern analysis. As we all know that at the crime scene, what we observe and what the investigators can see is the final stain on the wall, everywhere, and most of the stain already dried or almost dried. And what the investigators want to really know is based on the stain on the wall, they want to know like what kind of drop size before the impact on the wall and how fast these drops impact on the wall. And also they want to where they come from, right? The direction of these drops before they impact the wall. So with this information, because they can identify the position of the victims and also even find what kind of weapons used during the crime process. Therefore, in order to find those connections and establish the connection between the final stain and the impact conditions of the drops becomes extremely important here. So generally speaking, the impact of drops on different surfaces depend on many factors, and most importantly are the liquid properties, for example, like the viscosity, surface tension, and density. And the impact conditions of the velocity and initial diameter also very important. The surfaces, of course, will influence the outcomes of the impact, particularly if we have a solid surfaces or we have like textiles, for example, with absorbing and weakening happen for those surfaces. And at the same time, the ambient conditions also play an important role. And in the physics study and the droplet impact process, we like to develop some kind of dimensionless parameters like the Webb number and the Reynolds number to characterize the process. And in our later analysis, I will show you our correlation with those dimensionless parameters. In the BPA area, there have been a lot of studies on the solid and non-absorbing surfaces for bloodstains. And here, we just show you example, like if we have droplet with the size of D0 and a velocity U0 impact on surfaces with a 90-degree or perpendicular impact. And we can measure the final stain area, the diameter, as well as some kind of the number of the fingers formed, because for most of the conditions, we form some fingers because of the instability at the edge of the stain. So based on these two parameters we measured, we can actually establish some connection between these two measured parameters with the initial diameter and the velocity. So basically, we have two equations and we have two unknowns, and then a problem can be solved here. On the other hand, if we have a non-perpendicular impact, for example, with a certain incident of impact angle, and we do have another information we can find from the scene is the shape of the stain. So basically, we have three equations, we have three unknowns, so problem also solvable. However, when we go to the textile surfaces or fabrics with a lot of fluid absorption and weakening happened, and the stain becomes highly distorted, and all the stain shapes most of them becomes like very irregular. And also you can see here, for example, you have a stain right after impact and after it dry or after the weakening. There's little connection between these different stages. So because of this complexity and the distortion of the stains, the interpretation of these bloodstains on fabric surfaces becomes extremely difficult. Previous studies, like both numerically and experimental work, has shown the contribution of post-impact weakening on the final stain. And under a low impact speed, we can see that we form a small impact stain, and the waking after that will actually contribute a lot to the final stain. And under a higher velocity, the impact stain becomes bigger and the contribution of the waking actually weaker. But we also see that the shape becomes more irregular for a higher velocity impact. And experimentally, Dr. Michelson already did some work on the post-impact waking effect on the stain formation process. So first we have the impact process, we have a spreading and then retraction. After that, we have a little bit of a stable period with absorption there. After absorption, we actually have very strong waking, and we can see that the final stain area becomes much bigger than the stain area right after the impact. On the other hand, they also developed some kind of correlations between the stain area and the volume of the blood drops, and there's a good correlation there, and we can actually use this correlation to find the size of the drop using the area of the stain. And next, we want to present some of our research work, and our research actually focus on to really examine the formation process of stain on textiles by using advanced visualization system, and try to provide more detailed information on this process. And the fluid we use is actually blood, and the blood comes from like a porcelain whole blood. So we measure the density, surface tension, and the viscosity, and in our lab. The surfaces we used textiles include like plain woven fabric, and also twill fabric, as well as jersey knit fabric. What we show here is like we characterize the structure and the roughness of all the fabrics, and we also measure the yarn properties of all the fabrics. This can be used in later analysis. So a unique contribution of our research is like we develop advanced visualization system. And in this visualization system, we can capture the entire stain formation process from the very beginning to the very end. So what we use here is we have two high-speed cameras. We have one horizontally located, the other one like tilted with a certain angle. And these two high-speed cameras will help to capture the dynamic impact process. And then we have one digital SLR camera to capture the waking process. This is like in relatively low speed. And then we move the stain, the formed stain, to another digital camera, and then we can capture the stain after a certain long period of time. Finally, we put the stain under a microscope to look at the detailed structure of the dried stain. So with this complete information, so we can get different aspects of the stain formation at different stages. So here we try to show you like a sample data set we took for a certain conditions. So here, first is the high-speed videos will help us to identify the dynamic impact process, both horizontally and with the tilted angle. The tilted angle gives us a complete picture of the stain. And then we'll also use the slow camera to record the waking process. As you can see that the waking can be recorded for a relatively long time. Then we'll use digital camera to capture the stain after 5 minutes and 24 hours after the impact. Finally, we put this under the microscope, and then we can take images of the stain at different areas of interest, so we can compare the different conditions for later analysis. Now we can share some of the result and give you some feel about what really happened when the blood droplet impact on different surfaces. So first we want to show you the dynamic impact process. We have the impact velocity effect. And when the impact speed is relatively low, and you can see that after the drop touch the surface, and it start to spread and then form some craters as well as some rims on the edge. After it reaches to about its maximum spread, and then the drop will start to retract, and then we will finally form a pool of blood on the surface. At the same time, after you form the pool of blood, it's already get into the fabric by absorption, and also the wicking already start. Now you can see that the stain becomes bigger and bigger with time. Another thing I want to notice is under this low speed impact, we do not have any fingers formed at the edge. However, when we go to a higher speed, like we call this medium speed impact, we see significant amount of splashing here, and also a lot of fingers will form during the spreading process. And for these fingers, they can pretty strong, and some fingers will break up to small droplets, and some fingers will actually retract back by the surface tension, and then combine with the main stain. And all the separated droplets will fall back onto the stain, onto the surfaces, and then we'll start to get into the surface and get the wicking. Also, the main stain also start to get into the surface, and wicking already started. And the time here is pretty fast. It's a short period of time. And then we actually try to quantify the stain area during the entire process. And here I show you the spreading curves for a typical set of conditions. So we have pretty much the same diameter of blood drops, but they have quite a different impact speed. Here is like we use the slow camera, and we get the stain area. This is right after the impact. We can see that the stain area quite different for different speed, and we can see that higher velocity give us a higher stain area. And after a certain time of wicking, and we can see that after wicking, we will see most of the surface will reach almost the same area, and the difference becomes really small after the wicking process. So by combining the high-speed data and the slow-speed camera data, so we can actually classify different phases for the stain formation on fabrics. So first, you can see that we have this initial impact with the spreading, and then after we reach a peak, and then we will start to retract. After retract, we got a kind of relatively stable period with absorption of liquid into the fabrics. After that, the wicking will start. But the first wicking period is very fast wicking. We call this first stage wicking, followed by a second stage wicking, kind of slow down, finally reach a constant area, does not really change with time. So this shows us the entire process for certain conditions, of course. And if we put more analysis to look at the stain areas, particularly the stain area after wicking and before wicking, we call this SF and SI. And generally, we like to define some dimensionless parameters. We call this stain spread factor. We just want to use these dimensionless factors to correlate with the wave number we mentioned before, and see if we can find some universal scaling equations there. So what we find is when we look at the spread factor for the final stain, you can see there's little dependence on the Weber number. That means it's pretty much constant for all the impact speed. However, the beta I actually had a strong correlation with the Weber number, and you can see that the stain area before wicking kind of increased with the impact velocity. Also, we also did a NOVA test for the final stain spread factor, and we can see that we do have very little dependence on the impact velocity. So the final stain area is of great interest to us because this is what we actually observed at the scene, right? And here we already show that for different impact speed, the final stain area almost the same. So that tells us that the final stain area is actually independent of the impact velocity. This is not good from the forensic sense of view because we cannot find the impact velocity based on the final stain. However, the final stain area does have good correlations with the droplet volume or the volume of the blood droplets. But this also depends on the structure of the fabric. And here our data show pretty good correlations for plain woven, twill, and the knit fabrics. And based on this, we can actually using the bloodstain area to find the blood size. And the other interesting parameters we also want to say is like the spread factor or the stain area right after the impact. This is like pretty much close to the outcomes for solid surface impact. And in the literature, generally we will have a universal scaling law we can use for solid surfaces. We try to apply this universal scaling law to our data and also to some other data for fabrics. But we do find that this scaling law does not really give us a very good scale for all our data points. So we propose some modified scaling laws in our paper so we can actually fit our data pretty well with the modified correlations. The other important analysis we try to do is like we want to really look at the wicking contributions to the final stain, because we already show you that the final stain has little dependence on the impact velocity, and but the stain right after impact has a strong correlation with the velocity. Here we define a ratio like the difference of the spread factor divided by the final stain spread factor. And we do find that this one will become smaller when we increase the Weber number or increase the impact velocity. So we also did some asymptotic analysis of this, and we do find that we have some theoretical derivations there, and we do find that under very low speed, it approach to a constant value, and when we go higher speed, and we can have a good correlation with the Reynolds number here actually. So as we have demonstrated in the previous discussions, and the wicking actually contribute significantly to the final stain area. And based on our data, we want to develop some kind of simple models for the wicking dynamics. We want to see how the wicking will change with time. And based on our data and some theoretical analysis, so we actually propose this simple form of the wicking dynamic model. So in dimensionless form, this model we call it kind of semi-empirical because we can have some derivations and then followed by the correlation from our experimental data. Our data actually follow very well with this 1/2 power index here. So but the constant or the coefficient before the power function term actually depend on the Weber number. And we can see that we have a good correlation of this coefficient C with the Weber number. And this tells us that when we increase the impact speed or increase the Weber number, this coefficient becomes smaller. And this also tells us that for a higher speed impact, the wicking becomes weaker. So now we can summarize kind of the impact velocity on the stain process. This is particularly for the plain woven fabrics. So first we will say that the stain right after impact will increase with the increase of impact velocity, but the final stain actually independent of the impact velocity. We also find that the contribution of wicking becomes weaker at a higher impact velocity, and also the wicking dynamics we can model by a simple power law function, and the coefficient depends on the velocity, and it actually decreases with increase in velocity. And also this power 1/2 can correlate well with our data. So next, I think Dr. Michelson will talk about some other aspects of stains and also talk about the effect of fabric types.
Thanks, Tigong. The key things that a analyst does at a scene So looking at the stain shapes, the locations, and the stain sizes. And so Qigong showed us that on the three fabrics that we looked at, plain woven, a twill, and a jersey knit fabric, that the size of the stain after approximately 5 minutes is stable and tells us about the volume, or the diameter of the drop, or the volume of the drop. But it doesn't say anything about the shape, and the textiles can alter the shape tremendously. So an analyst likes to look at the shape because that often tells us the direction of motion of the drop. And so we're going to look at that next and see what we can do with that. So first off, we can, here's two stains, one a low velocity stain and one a high velocity stain. The low velocity stain, we can see the initial impact with this bright red area here. It's quite small, pretty much circular, pretty symmetric. While at the higher speed, we see that the bright red is, the edges of it are quite pronounced. There's fingers and so forth around the perimeter. The stain itself at this point is much larger. As time goes on, the first stage of wicking that Qigong mentioned is this darker red region. We can see that that initial drop on the small side spread out a very large amount, and the edge, the perimeter of that, began to get a bit rougher. While on the at the high speed, what we see is it didn't really get much larger. The dark red is only a little bit larger than the bright red. And a lot of the fingers that we saw before are beginning to be hidden by the spreading, by the wicking blood. And then the pink area on both we can see is a little bit larger. That's after the second stage wicking. And at the low velocity, the perimeter now has become quite rough. At the higher velocity, it has become less rough. It's still more rough than the low velocity, but it's much less rough than the initial. So much of the initial information is covered by the spreading droplet. We can look at this as a function of angle, and we'll see that these angles relate to the direction of the yarns in the fabric. And so we can look at these to get the aspect ratios. This particular plain woven was balanced. The types of yarns and the number of yarns in both directions was the same. So we would expect on this fabric to have a nice circle. On other fabrics, even with a 90-degree impact, we may have an elongated, an elliptical stain, simply because of the fabric structure, not because of the dynamics of the impact. And we can see here that as we increase the velocity, the aspect ratio remains close to 1 on this particular fabric. We can also do an analysis of what's called the irregularity factor, which is just a measure of how close this stain's shape is to a perfect circle. And so the larger IF, the further away we are from a perfect circle. If we look at the impact velocity, we can see at our very low velocity, our first bar here is right after impact, and we can see that has an irregular factor of very close to 1. And with time, it becomes less regular because it's wicking into the structure, and that's controlled by the wicking properties of the fabric. At the other end, at the very high velocity, we see that the irregularity factor initially is very high, and the wicking actually reduces that irregularity. The final irregularity is still higher than that of the low velocity, but a lot of that difference is now hidden and missing. If we look at the fabrics themselves, we can see the plain woven, the yarns are the same size and the same number of yarns per distance in both directions. Front and back of a plain woven are the same. So this is pretty standard. If we go to the twill, we see that the size of the yarns, the number of yarns are different in both directions, and the front and the back of the fabric are very different. For the knit, likewise, the fabrics, the front and back are quite different. And if we look at the yarn sizes, we'll see for the plain woven that the size of the yarn, as we measure it by denier, are essentially identical. In the twill, the yarn sizes are different by a factor of 2 in the two different directions, and the number of yarns are also different. And the jersey knit, there's only one yarn, and its size is between that of the twill and the plain woven. So all of these go into affecting the stain. We're not going to talk about those details today, other than say that plain woven, where we're getting a beautiful symmetric initial impact, and the wicking is still fairly round, is because the yarns are the same in both directions. We should not expect that in general. Now, when we look at the data that Tigan got, we see that in our non-splashing conditions, after this is after only basically 1/100th of a second, the impact event is over. We've got a couple of fingers here, but they're quite short. At high speed, we see the perimeter is much, excuse me, all of these are low speed, sorry. We see the perimeter of all of these are fairly smooth with very, very few fingers. When we go to the splashing conditions, high impact velocities, we see here that, again, about 1/100th of a second, we see lots of fingers, and we see lots of these little drops, these satellite drops being thrown out from that initial stain. The same thing happens on the front and back of the twill, the front and back of the knit, with lots and lots of secondary or satellite spatter. If we go back to Tigan's analysis, we see for all three fabrics and both sides of the twill and the knit fabric, the impact-induced stains increase approximately with the log of the Weber number. Nice, consistent, behaving quite similarly, where the final stain diameter is independent of the Weber number for all three. However, it is dependent on the fabric structure. So our knit, the final stain factor is much smaller than on the plain woven, but it does not depend on the velocity. This is the same the same semi-empirical equation that Tigan had, and the final empirical where we allow this exponent to vary. We'll see why in a second. You can see the curve fits are pretty good. But we'll see that here for the knit, this exponent is not a half, it's a 0.9 in this case. And so that doesn't fit the semi-empirical equation. It has to be modified to take into consideration the knit. And we can see that here also that the plain woven, the twill, behave classically, where the knit is wicking much faster than what we would expect. But that also means the duration of wicking is smaller for the knit. And here again, we get the same behavior as we change our Weber number, with the power coefficient remaining approximately constant for most fabrics, but for the knit, it's actually decreasing as we increase. So what we're seeing in the wicking rate, if we were standing there watching watching everything occur immediately as the crime is happening, or the bloodletting event is happening, we would notice these changes. But remember, we're coming after typically an hour, 2 hours, maybe several hours after the crime occurred. Some other interesting things we can get from the splashing dynamics is we can get the number of fingers and the number of satellite drops from the initial impact. And so the number of fingers on these fabrics, these are all under the same condition, that the number of fingers is about the same regardless of what our fabric is, where the number of satellite drops change tremendously based on our fabric. And so this may give us an opportunity to better understand the bloodletting event. And so when we take different size drops, different fabrics, and different drop heights, we can get our splashing parameter, tells us how much is splashing based on our experimental conditions. And the important thing is the number of drops, number of fingers, when we look at our high speed, depends nicely on the impact velocity, where the number of fingers, well, also strongly, but a different dependence than on the number of fingers. And so when we combine all of this data, we find that the number of fingers is approximately equal to 0.7 times the square root of the Weber number. So that's what we're getting here over quite a wide range in Weber numbers, more than an order of magnitude. But for the number of ejected drops per finger, we get a somewhat different dependence. For the knit and for the woven, plain woven, we get this behavior, a nice, more or less straight line with Weber number, where for the twill fabric, the number of satellite drops is approximately half the number of fingers. Now remember, the number of fingers is proportional to the square root of the Weber number, where the number of drops per finger is either independent of Weber number for the twill or linearly dependent for the knit and the plain woven. So when we combine this information, we can now get information about the drop when it impacts on the fabric. So the number of fingers is, as I said, 0.7 times the square root of the Weber number, but this is the number created in the impact. What you observe at the crime scene is much, much less because, as we saw earlier, the wicking spreads out and covers many of those fingers. So the number of fingers you observe is far, far less. You may not observe any fingers whatsoever, even though they may have been created during the impact. However, the number of ejected drops, if the drops are thrown far enough away so they get outside of the wicking diameter, then we can measure the number of ejected drops, and those, just plugging into our relationships, give us the number of ejected drops is about, for the twill, is about 0.35 times the square root of the Weber number. And for the plain woven and the knit, this number looks very small, 0.0013, but it's times the Weber number to the 3/2 power, so a much stronger dependence on the velocity. The number of observed ones is less than the actual number, but since the satellite drops tend to be thrown a fair distance from the parent stain, we will see most of the ejected droplets at moderate speeds. Important point, more work is needed to verify these results and to make it more general for other types of fabrics. Each fabric has its own dependence. But this says there's the potential to get more information about the event from the ejected drops, the satellite drops, than from the parent stain. The other thing that the satellites do is help identify the direction from which the drop came, since the satellite drops tend to go in the direction of the moving droplet. So combining this, we can get the D0 value, the diameter, the drop size from the total stain area. From the number of ejected drops, we can get the Weber number, either for our twill or for our plain woven or knit. And we know what the Weber number, by definition, the density of the blood divided by the blood surface tension. So we know those. D0, which we just measured, and so that gives us our velocity. And so it may be possible, by looking at the ejected drops and looking at the stain size, to extract the velocity and size of the drop together. Again, as I said, more work is needed to verify this, but it points to potential of analyzing stains on fabrics in more detail than what has been possible in the past. And so in conclusion, all the tested fabrics, we looked at the impact velocity effects and the stain factor right after impact and the wicking contribution in forming the stain area. The impact velocity has a very weak influence on our final stain factor or the final stain diameter. On different fabrics, the wicking dynamics and splashing dynamics can be quite different, but there are some underlying things that we can discover, as we just mentioned, with the number of ejected drops, the total size. The bloodstain shape analysis, we get two new messages that the bloodstain aspect ratio can be larger than 1. Depending on the fabric, it can be much larger than 1. For the fabric, for the plain woven we looked at, it was very close to 1. It varied a little bit with impact velocity, but was still fairly close to 1. The irregularity factor, likewise, can be quantified to get more information, but remember, this is after wicking. Much of the information we're interested in has been eliminated by the wicking process. And then the relationship between the observed stain area and the airborne blood drop volume was studied in different textiles. Each textile will have a different behavior, but once we understand that behavior, then we can evaluate it as a non-destructive approach to getting blood drop volume and potentially the velocity. Tigang and I would both like to thank you for your attention, and I think the next step is opening for questions.
So thank you very much for both presenting today. Much appreciated and fantastic presentations. And we will start off with our first questions. And our first question of the day is, are hand-woven fabrics difficult to analyze?
I'm not sure what's meant by that question. So is that the structure of the woven fabric or the bloodstain on the fabric?
I'm not sure. We'll move on to our next question then. Was any research done on droplets that reached terminal velocity?
For our research, no, we didn't reach the terminal velocity, and not that high. Yeah.
Next question. With the different variants of fabric that have to do with microbiological functioning, such as antimicrobial, are there complications with this method if the type of fabric is the one to be analyzed, if this type of fabric is the one to be analyzed?
So there's two aspects there. First off, the studies that we did on very carefully controlled fabrics because we needed to get down to the fundamentals of what's going on. Most fabrics that you will come across have, other than simple t-shirts, have some sort of finish on them. That may be a biological antimicrobial, it could be anti-stain and so forth. Those don't have much of an effect on the dynamics because the dynamics are really over with before there's much interaction with the surface chemistry. However, it will affect the wicking somewhat. It might affect the wicking somewhat. But that can be compensated if you put on a drop of water. You'll see a lot of, just literally lay a drop of water on, you'll see a lot of those effects in the wicking. So we're a long ways from being able to answer that type of question, but from what we already know from textile research, that is a relatively small contributor. It affects the overall stain diameter, but less on the shapes and so forth.
And one question from the audience, were the studies done on new fabric or washed fabric?
Everything was washed at least 5 times by industrial laundering, well, industrial processes that are much more vigorous than your home washing, but they were not used fabrics. They were not dirty fabrics. Those can have effects on the detail of the shape. These would apply to areas that are not particularly dirty, that every garment that is worn, there will be areas with less dirt and more dirt. This is the less dirt region.
Speaking of dirt and washing, another question would be, are there any further studies that are being done on the age of fabric? So adjusting the number of washings and then chemicals within the fabric or dirt on the fabric?
So there have been some studies. When people do laundering on textiles before they're doing their tests, it's really to stabilize the structure and to remove any processing chemicals along the way. There is not much done on that, and part of it is that we're just beginning to understand the interaction of the blood with the textile and the effects. And so it's too early to say, okay, if we put this dirt on it, this body oils and so forth, any lotions or creams and so forth, those are details that that will certainly complicate the issue and may render some of this inappropriate. But until we understand the base clean textiles, we don't really have much hope in understanding the dirty textiles.
Thank you. Our next question, can you please re-explain or go into further detail regarding the correlation between the number of fingers, the number of satellite stains, and the conclusions being drawn there?
Okay, so this is preliminary result. When we look at the data that the Tiagong showed us, we see that the number of fingers is controlled by the rheology. So the surface tension, the viscosity, and the impact velocity. Those are pretty standard. You saw from his images, it doesn't matter if it's a drop on a textile or a drop on a piece of concrete or anything in between. So the number of fingers is due to the rheology. However, the number of cast-off drops, ejected drops, is also dependent on that. However, the wicking depends on the textile structure and the surface properties of the textile. So they separate in that sense. More work needs to be done. What we came up with is based entirely on those curve fits. They have not been tested for a larger scale study to say, yes, this is definitely true of all textiles. Remember, there's 100,000 different textiles. We studied 3. So we're starting to understand it. We're starting to, understand is too strong. We're starting to pull apart the pieces that are important to understand it. We're not there yet. but we're moving in the right direction. There is information you can get from the stain on the textile, but what you've learned in your courses of do not try to get a angle of incidence from your blood stain on textiles. We would agree, do not try, because the wicking dominates that process, not the impact event.
Excellent, thank you. And then we have a couple questions about types of fabrics. Were your fabrics made from natural or synthetic fibers? And a follow-up question would be, have you done any work on the differences between these two types of fibers? Okay. Sorry.
So all of our materials were 100% cotton. We're trying to limit the number of variables. The difference between natural and synthetic, you have to be careful because most of the synthetics, or many of the synthetics, are cut to staple fibers, and a big difference is between staple and filament. There's also the difference in the surface energy between the different types of fibers. I would not use natural versus synthetic in the sense that identifies it. It's really much more complicated than the source of the fibers. We could do 5 webinars just on that. So more studies need to be done. Clearly, textiles are clearly a very important aspect of bloodstain pattern analysis, of bloodstains at scenes, but we're a long ways from understanding that.
And then have you looked at the dampened or wet fabric to simulate the wicking differences? For example, if someone happened to be sweating during a blood shedding event.
So no, again, that's added detail that the water part of sweat goes into the spaces between the fibers within the yarn. The red blood cells might go into that. They may be excluded from that. So if you have water already in those regions, that's going to greatly alter the wicking. One would expect, based on that, larger stain areas, but at the same time, more difficult to observe because it's being diluted by the sweat and dirt and so forth. So no, we have not looked at that. I'm not aware of anyone who has looked at that because nobody understands bloodstains on textiles very well. So we're starting at the easiest and working towards the more complicated.
And then I guess that would tie into the next question, which would be, how would the dilution of blood change your results? And I'm assuming that would need to be further studies or...
So if the blood is more dilute, then the stain is going to be bigger if it's diluted before it goes on. So it's a question of when does the dilution occur? If it's before, there's more liquid, so the stain will be larger. But when we're saying that it does not, that the area depends on the drop size, that's for the fabric, but it's also for the, if you have different hematocrits, which means different dilutions, that will also affect the extent of wicking.
Thank you. Our next question is, this model seems to be a moving drop hitting a fixed fabric target. Any thoughts on moving drop hitting a moving fabric target?
Maybe I can comment a little bit on this. So generally, when we talk about solid surfaces, so we say we try to use the relative velocity, but this is, I think, even you use the relative velocity, there is still some difference between those two scenarios because the boundary layer, when the object is moving, there will be established a boundary layer on the surface. So that interaction with the droplet will be different, even though sometimes we make the assumption they are the same. But for fabrics, I also expect it can be very different, even though you can assume at the very beginning, relative velocity, but I think we didn't do some on similar work on fabrics, but we started to do some work of droplets interaction with the moving surfaces. We'll see how that goes, but I think for fabric, that will be different, I think. Yeah. That's my opinion. I don't know how Steve thinks.
So yeah, I think if it's at a very low velocity, when you're walking, your velocity is relatively low. So there, the boundary layer will not be as important. But if someone is running, sticking their arm out of a window as they're driving along, those certainly are going to have unknown effects. The boundary layer will become quite critical in those cases.
Thank you. Next question. Caller would like you to speak about the speak towards discerning spatter from transfer on fabric and the associated limitations.
So I see this one came from a good friend, Kevin. I hate you, Kevin. No, when you look under the microscope, a transfer is applied to one side with essentially zero velocity. The wicking will start from that side. If it's a limited, say a spatter stain where you have very small drops, you would not expect very much motion of those. At the same time, when you impact at higher velocity, there is the potential for the drop to penetrate into the structure further. However, I don't think anyone has a good, I have not seen anything published where there's really a good definitive answer to that question. I know that's one of the holy grails in bloodstain analysis on fabrics. Transfer is critically important, and distinguishing between the two is critically important. Under a microscope, you may be able to see things, but we have not really done enough work on that to be able to answer that.
And could you explain why the final stain area is so different for all three different fabrics?
That one's easy. The yarn in each one is different. The construction of the yarn is different. The construction of the fabric is different. So the size of the stain is largely due to how much open space there is in the three dimensions. So if you have a thick fabric, it can absorb all the blood locally. It doesn't spread very much. If it's a thin fabric, it spreads out further to get the same amount of open space. Each yarn type depends on the fiber size, the twist, the yarn size, all kinds of details of the construction that we've begun to probe but have not gone very far in that yet. But all of those affect the ability, they affect the open space within the textile, which is where the blood goes. So your sportswear is designed to spread liquid out quickly and a long distance. So there you would expect a large area for the same volume as a raincoat that's designed to prevent spreading of the liquid would have a very small one. So those have to do with the details of the fabric construction. We understand from moisture management in textiles for our sportswear and other clothing a lot about that. That has not been looked at extensively or probably at all in the published BPA literature. But we understand qualitatively what's going on. Quantitatively, I'm not sure we're there yet.
And one of our last questions the caller says, in BPA training, they are taught that they cannot determine the direction of flight from bloodstains on textiles. Are you saying that it may be possible to determine the flight direction?
If you have satellite spatter then the direction of the satellite spatter may tell you something about the direction of the impact. If you're looking at the parent stain, you should not ever, in my opinion, use that to try to interpret the direction. And that's what they're really referring to in the courses, because they're looking at the primary stain. The primary stain can tell you exactly the wrong thing. But if you look at the satellite spatter, that is not affected by, it's only slightly affected by the textile structure. And so the flight of those drops have the potential. This is something that I just, as we were preparing these slides, I said, I think we can. But it's from the satellite spatter, not from the parent drop. And so the courses that you're being taught is correct, but it may have a little wiggle room when you have higher speed impacts and you have satellite spatter.
Thank you both very much. Unfortunately, that is all the time we have for questions today. I would like to thank both Dr. Mickelson and Fang for your presentations today and your ability to be with us. Excellent presentations as always, and great audience participation as well. Thank you again so much.
Thank you so much. Thank you all for joining the webinar.
Yes, thank you. And we are available for questions via email. They're on the screen, so don't hesitate to send us questions. Don't expect an answer tomorrow because this is coming into the holiday season, but we will try to answer your questions as best we can. And probably it's worth copying both of us on those questions. Higang is our rheologist, our fluid mechanics person. I'm the textile person. Together, we should be able to answer, or we should be able to address most of your questions, maybe not answer.
Thank you again.
As you depart, we kindly ask that you take a minute or two to complete our post-event questionnaire. The link will be pasted in the chat and also appear as you exit the Zoom room. Your feedback helps us improve our outreach efforts and continue to provide the best experience possible. Thank you again for attending, and we hope everyone has a great day.